Whenever you scrape your knee or get a paper cut, those injuries leave an opening to your body that must be covered up. And one thing that has to occur for this healing is the regeneration of cells, which is done through a process called mitosis. Mitosis is a 5-step process that cells go through to create a clone of itself. In this video, I will be walking you through those 5 steps. But before we actually get into this mitotic phase of the cell cycle, I'm going to walk you through the preliminary processes as well. We begin the cell cycle with a process called interphase, which is usually much longer than the mitotic or M phase, where the 5 steps I talked about before occurs. In fact, interphase accounts for about 90% of the cell cycle. Interphase can be divided into 3 different phases called the G1 or first gap phase, the S or synthesis phase, and the G2 or second gap phase. These G or gap phases were originally named gaps when they were first observed because the cells appeared inactive, but now we know that intense metabolic activity actually occurred during these gap phases. As a whole, you can think of interphase as the preparatory phase for the cell to duplicate its components before it actually physically divides. Interphase begins with the G1 phase, where metabolic activity and growth occurs in the cell. Then, DNA duplication and more metabolic activity and growth occurs in the S phase. Finally, the cell goes through one last process of metabolic activity and growth before it actually enters the mitotic phase of the cell cycle. Now that we've covered interphase, let's talk about the actual mitosis. The 5 stages of mitosis are called the following: prophase, prometaphase, metaphase, anaphase, and telophase. Overlapping with the latter stages of mitosis, cytokinesis completes the mitotic phase. Before we go more in depth with each of these 5 stages, I want to talk a little bit more briefly about mitosis and what each stage's prime function is. The steps of mitosis is primarily about dividing DNA accurately and safely, so each step will be focused on doing things with the genetic material of the cell. In prophase, the chromatin fibers in the nucleus become condensed into chromosomes. In prometaphase, a mitotic spindle forms and attaches its arms onto each chromatid. We'll get into what these terms mean later on. In metaphase, these chromosomes line up in the middle of the cell, ready to be separated. In anaphase, the chromosomes are separated. In telophase, a process called cytokinesis occurs where the cytoplasm of the cell is divided into 2, which makes it clear from the outside that the cell has physically divided. Now that we have briefly looked at the process, let's actually dive deeper into each of these steps. First, let's roll back a little and take a look at the end of the G2 phase to see what the cell looks like right before it enters prophase. Some notable characteristics include the fact that a nuclear envelope encloses the nucleus. The nucleus contains one or more nucleoli. Two centrosomes have formed by duplication of a single centrosome. Two centrosomes have formed by duplication of a single centrosome. If this term seems unfamiliar, here is the definition of what a centrosome is as a refresher. So centrosomes are regions in animal cells that organize the microtubules of the mitotic spindle used to divide the duplicated chromosomes. Each centrosome contains 2 centrioles. Chromosomes duplicated during S phase cannot be seen individually because they have not yet been condensed. Now let's actually take a look at the beginning of mitosis, which is prophase. So in prophase, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes observable with a light microscope. The nucleoli disappear. Each duplicated chromosome appears as 2 identical copies joined at an area called the centromere. We call these copies sister chromatids. They are stuck onto each other by special proteins called cohesins located at the centromere. The mitotic spindle begins to form. It is composed of the centrosomes and the microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosomes are called asters. Asters mean like star, so you can see in this diagram how the arms are like stretching out. And it kind of looks like a star, which is why they're called asters, just as like a little side note. And then lastly, the centrosomes move away from each other, propelled partly by the lengthening microtubules between them. Now let's move on to the prometaphase. So during this phase, the nuclear envelope fragments, and the microtubules extending from each centrosome can now invade the nuclear area. The chromosomes have become even more condensed. A kinetochore, a specialized protein structure, has now formed at the centromere of each chromatid. This means that 2 form per chromosome because there are 2 chromatids per duplicated chromosome. some of the microtubules attach to the kinetochores, becoming kinetochore microtubules, which jerk the chromosomes back and forth. Non-kinetochore microtubules interact with those from the opposite pole of the spindle, lengthening the cell. Next is metaphase. During metaphase, the centrosomes are now at opposite poles of the cell. The chromosomes have all arrived at the metaphase plate, a plane that is equidistant between the spindle's two poles. And that means like the distance between this pole over here and the metaphase plate, and the opposite pole and the metaphase plate is like same. So equidistant, same distance from the metaphase plate. So you can basically think of the metaphase plate being like right in the middle if we imagine like the cell as like a circle, like in this diagram. Okay. 1 second. And then for each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. So now anaphase comes next. This phase is the shortest stage of mitosis, often lasting only for a few minutes. So it begins when the cohesin proteins that we talked about before that keep these sister chromatids together are cleaved. And this allows the two sister chromatids of each pair to part suddenly. And each chromatid thus becomes an independent chromosome. So the two new daughter chromosomes begin moving toward opposite ends of the cell as their kinetochore microtubules shorten. Because these microtubules are attached at the centromere region, the centromeres are pulled ahead of the arms, moving at a rate of about 1 micrometer per minute. The cell elongates as a non-kinetochore microtubules lengthen. And by the end of anaphase, the two ends of the cell have identical and complete collections of chromosomes. So now, lastly, comes in telophase and cytokinesis. So here, the two daughter nuclei form in the cell, and the nuclear envelopes arise from the fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. The nucleoli reappear, and the chromosomes become less condensed. So before we had everything purposely condensed so that it's easier to like separate, but it becomes less condensed. And you know how chromatin fibers in normal cells, like inside your body right now, that are non-dividing, are usually found with like all of their genetic content to be like all dispersed and disorganized. So it's going back to that since division is coming to an end. And any remaining spindle microtubules are depolymerized. And cytokinesis is a process of dividing the cytoplasm into two through a formation of a cleavage furrow, which pinches the cell in two. And this process is usually well underway by late telophase. And so after all of these 5 stages, mitosis officially comes to an end. If this video was helpful, please leave a like to let me know that this type of content is helpful. And if you'd like to get notifications whenever new biology content like these are posted, please consider subscribing. And lastly, if you have any questions or thoughts, feel free to leave a comment and I'll usually respond within a day or two. Thanks for watching and happy studying. Thank you.